I'll be discussing about radioimmunoassay, which is a very sensitive assay which can detect the antigens or the antibodies at the nanomolar level. To perform this assay, highly specialized, sophisticated laboratory is required because the radioactive isotopes are going to be used for labeling the antibody or labeling the antigen. Since these radioisotopes emit radiation, the radiation is harmful for the human beings. Therefore, the laboratory should be in such a condition that it should be protected and easy access is not available for others. Only certified person who is qualified to handle radioactive isotope is allowed to work in this lab. Second, the laboratory has to be recognized for its safety measures by the International Atomic Energy Commission. Only after the certification of the lab is permitted to procure the radioisotope labeled material to be used for the diagnostic purpose. The most common radioisotope used for the labeling of antibodies is I-125 or I-131, that is iodine 125 or iodine 131. It is also used for labeling the antigens, and one of the important aspect of I-125 or I-131 is it is easily attracted towards tyrosine or has strong affinity towards tyrosine. If the tyrosine residues are present in the antibody or the antigen, the tyrosine residues gets readily labeled by I-125 or I-131. Therefore, it is called as direct labeling without involvement of any coupling agent. The antibodies or the antigens having tyrosine residues can be labeled with I-125 or I-131 with a simple chemical reaction. Whereas when yttrium or indium are used for labeling the antibody, a chelating agent is required called as diethylene triamine pentaacetic acid. In short form, it is called as DTPA, which is used as a coupling agent or a chelating agent, which will help in binding of yttrium or indium, radioactive yttrium 90 or indium 111 to the antibody, and it is called as indirect labeling. Radio labeled antibody or antigen, when it is bound to the target, it emits radiation, and that is measured by using scintillation counter. Let us take a simple technique here. Now, how radioimmunoassay is performed. In this example, HBsAg, that is hepatitis B antigen, is used, which is radioactively labeled with I-125. And the serum sample to be analyzed for the hepatitis B antigen is to be used. In the first left-hand side example, they have shown infected serum is added together with the hepatitis B radioactively labeled antigen to a container where the antibodies against hepatitis B antigen are already immobilized on the surface of the container. The antibodies are immobilized by using protein A which is coated over the surface of the container, and protein A attracts the Fc arm of the antibody. Therefore, the Fc arm gets anchored onto the surface of the inert surface, where the protein A is already coated. The immobilized antibodies will be readily available in the container, to which the serum sample of the infected person is added, together with radioactively labeled hepatitis B antigen. There will be competition between the unlabeled hepatitis B antigen, which is found in the serum, with the labeled hepatitis B antigen to bind to the antibody antigen binding site. And if the concentration of the unlabeled antigen is more compared to the concentration of the labeled antigen, more of unlabeled antigens will bind to the FAB site and the labeled antigen will not be able to bind. Similarly, vice versa, if the concentration of unlabeled antigen is less and the concentration of labeled antigen is more, that is radioactively labeled antigen is more of labeled antigen will bind to the antibody FAB site. In the second example, which is shown on the right hand side, the uninfected serum is there, that is the serum negative for hepatitis B antigen. Therefore, the FAB site of the antibodies which are immobilized in the container will be all bound with the radioactively labeled antigen, because there is no unlabeled antigen here. Now, let us take this example of the right hand side of the uninfected serum, and if you take the radioactive count of this sample, in the curve the sample will have the count of the highest radioactivity, which will be here between 60 to 70, because there is no competition for the labeled antigen here. Almost all FAB sites of the antibody are occupied by the labeled hepatitis B antigen. Therefore, highest amount of count of the radiation will be emitted, and there is no unlabeled antigen. It will be 0. So, it will be somewhere here. Now, as you increase the concentration of unlabeled antigen in the sample, the curve will appear like this, partly linear, and then it will start becoming flat. At one point, there will be minimum count of the radioactivity, and at that point the concentration of unlabeled antigens bound to the antibody will be highest. With minimum count of the radioactivity, the concentration of unlabeled antigen bound to the antibody will be highest. So, the part which is shown partly, that is somewhat linear, approximately linear, and that is the part to be considered for the finding out of the antigen concentration in the unknown serum sample, which is suspected to be having the antigen. The linear part is the area to be used for analysis, in the sense the serum sample obtained from the suspected patient to be analyzed for the antigen should be sufficiently dilute where the radioactive count falls in that linear range part, which is equal to the concentration of unknown antigen, which can be equated to the concentration of unknown antigen. So, radioimmunoassay is used for the most sensitive assessment of the very low concentration of the antigens, like the thyroid hormone or the HIV antigens like the GP120 protein of the HIV virus, like that. So when the antigens are in the low concentration in nanomolar level, the radioimmunoassay can be performed with approximate dilution of the serum sample. With trial and error, one can achieve the range that is shown here in the curve on the linear part, and then one can find out the concentration of the antigen in the unknown sample by comparing it with the radiation shown by the bound radioisotope bound antigen. We'll take another radioimmunoassay, which is radioallergosorbent assay. Why it is mentioned as radioallergosorbent assay? Because it is meant for detection of allergic antibody for allergens. Generally, allergic antibody is IgE in majority of the cases for most of the allergies against all allergic antigens. The major antibody is IgE. To some extent, IgG also may be involved, but to a less extent it is. Here in this case, in this example, on the inert surface of the container, the antigen, that is allergen, is immobilized. Let us say you take microtiter plate, 96 well microtiter plate, and to the protein A coated 96 well microtiter plate, you add different types of allergic antigens which you suspect that those antigens are responsible for the allergy in the person. So after adding each individual antigen to the particular well of the microtiter plate, incubate the plate for some time so as to facilitate the allergic antigen to bind to the wall of the container, that is to the well. After a sufficient incubation period, wash off by using the saline or a suitable buffer. Whatever the excess antigen that is there which is not bound will be washed off, but antigen that is bound will remain there bound to the inert surface. Now in each well where individual different antigens were added, you add now the serum of the patient who is suffering from allergy. To all the wells, the small quantity of the serum sample of the patient is added. Now the patient is supposed to have the antibody to those allergens, and that is called as first antibody or primary antibody, and generally it is IgE. And if IgE is present in that patient corresponding to that antigen, after adding that serum to the wells, the antibody will bind to the antigen. Now once again, after some incubation period, you give a thorough wash to all the wells with a saline or suitable buffer. The antigen-antibody complex formed is stable at a given pH and it will not be disturbed. And after this wash, you will see that if the antibody corresponding to that antigen is present, will have been bound and will be there in that well. To that sample now, to all the wells, add radioactively labeled secondary antibody, which is anti-IgE antibody, which is raised in an animal. Anti-IgE antibody is raised in an animal, and purified, and then it is radioactively labeled. And this secondary antibody, you add it to that samples, incubate it for some time, and then give a wash. If IgE antibodies of the patient serum is already bound to the antigen, the secondary antibody added, which is against the IgE, will bind to the IgE and will remain there. Now give a wash and then subject the samples for scintillation count. You will see that wherever the particular allergen or the antigen was bound by the patient's serum IgE antibody was there the radioactively labeled antibody would have been bound to it and would have started emitting radiation. From this, one can identify which are the allergens that are capable of causing the allergic reaction to that patient. Therefore, he has produced IgE antibodies in his serum. And those allergens can be classified and the patient can be advised to avoid those allergens to decrease the concentration of IgE in his blood so that his constant suffering from allergy can be avoided. Since the test is named as radioallergosorbent assay, the allergen, that is antigen, it's adsorbed onto the surface of the inert material, and then it is used for the detection of the antibody, allergic antibody produced in a patient. In another test, one can detect once again the antibody present in a patient by immobilizing the antibody on the surface of the inert material. That is, once again, you can use microtiter plate or a tube. First, the suspected patient serum having suspected antibody possibly is added. And before adding that suspected serum to the container wall is coated with protein A. And this assay is called as radioimmunosorbent assay. Immunosorbent assay because you are capturing the antibody on the surface, inert surface from the patient serum. The antibody that is captured will attach to the surface of the inert material through its Fc arm because protein A coated surface attracts the Fc arm. And FAB arm of the antibody will be free, available for binding of the antigen. If you are looking for a particular antibody corresponding to an antigen in a particular patient, and you have immobilized that antibody on the inert surface by adding the patient serum to the container, you have to add now, after thorough wash of the container, the antigen corresponding to that antibody should be added. The test antigen corresponding to that antibody should be added. And when you add the test antigen, if the antibody corresponding to that antigen is already immobilized, that antigen will bind to the FAB site of the antibody. Once again, give a thorough wash. The complex of the antigen antibody is stable at a given pH. After the wash, you add now the secondary antibody which is raised in an animal against that antigen, and that secondary antibody is labeled with radioisotope. On adding that secondary antibody, if the antigen is already bound to the antibody, that is primary antibody and available in the wells, the secondary antibody also will go and bind to that antigen. Now here, the antigen is sandwiched between the patient's antibody and the secondary antibody. The secondary antibody which is attached to the antigen will remain stable. Give a wash. Now subject the container of this complex for scintillation count. If the radioactively labeled antibody is already bound to the antigen there, it will emit radiation and one can count it. So this test also can be used for detection of antigen in the patient's serum by immobilizing an antibody which is available for that particular antigen, and that antibody is available in the pure form and it is immobilized on the inert surface using protein A, and the serum to be tested for a particular antigen should be added, and that antigen corresponding to that antibody, if it is present in the serum, which will bind to the antibody at the FAB site, and then you add a secondary antibody that is labeled with radioisotope corresponding to that antigen. In this case, you are detecting the antigen in the serum sample by prior immobilization of an antibody corresponding to that antigen in the inert surface. Both ways, radioimmunosorbent assay can be performed, either to detect the antibody in the patient's serum or to detect the antigen in the patient's serum. And this technique is highly sensitive, as I mentioned. Antigen levels up to nanomolar level can be detected in the serum sample. Hence, this technique is used for detection of very low concentration of antigens which cannot be detected by other techniques. Thank you.